So we'll move on to lipid as another biological molecule. So you want to, as I would normally advise, pause the video first to see if you can answer the question, and afterwards you play this. So like this floating part tab is the answer to this question. So general structure of a fatty acid is RCCOOH. So what does this represent? Pause the video and think about what that means. So that is the carboxylic acid group of the fatty acid structure. Okay. Then the next question says, figure 1 shows the structure of fatty acid group. The R part, which is always the variable group, or the hydrocarbon group part of it. So what type of group is R? So what do you think this R means? What does that mean? Then explanation to what you think. Now please pay attention. It says the type. Take note of this structure. Take note of any unusual thing you saw there. If you've done this will just click again. So pause the video and see if you can answer. So this is an unsaturated fatty acid. You remember I said the type because of the fact that it's unsaturated because this doesn't have hydrogen, so it's not fully occupied. That's what it means, like it's unsaturated, and that's why I have double bond here. Okay, now, and this is because of the presence of carbon to carbon double bond. AQA specifically demands that you mention carbon to carbon double bond when you're mentioning anything that is unsaturated. This is very important. Okay, moving on. The next question says, describe how you will test for the presence of a lipid in a liquid sample food. So the food is already liquid already. So and how you test for it? How many marks? 2 markers. So think about this real quick. Lipid sample is already in liquid form. So you add ethanol to the sample, then add distilled water. AQA usually want that distilled water. Sometimes they may waive it, but there's no harm in giving them the exact that they want. Then shake. If lipid is present, then this should be then a white emulsion will appear. Please, it is not precipitate. It is emulsion, because precipitate is a solid. This will be a solid, just going to be some sort of emulsion. Okay, please pay attention to those keywords. Now the next one says, describe how a triglyceride molecule is formed. Triglyceride. I'm emphasizing that try for a reason. 3 markers. So how will it be formed? I'm going to break out your points into 3 points, or we answer to 3 points, so to say. Pause the video and see if you can attempt that. There you go. So this is formed by 3. You remember I was emphasizing the 3 fatty acids and a glycerol molecule. Now these are joined by an ester bond. Okay, then 3 water molecule is released in this condensation reaction, because if you are adding, the moment we are joining, there will be water formed, and here we say we have 3 fatty acid. So that basically means that we have 3 water molecules being formed in this condensation reaction. Each of those points is a mark, and you have to make sure you say them sequentially in the right order. However, it is possible for you to draw, say for example, you draw your glycerol like that, then you show us that we have fatty acid 1, fatty acid 2, fatty acid 3, then you tell us plus water, 3 of those, and you have to tell us this is ester bond. You can also get the same mark doing that. Either way, that's fine. Okay, the question says, table 1 shows the properties of 4 fatty acids, and these are the properties here, and figure below shows the diagram of this fatty acid. So I'm going to keep this here for you, this second table here, which is on the answered one, and you look at this. So you're meant to use this diagrams to, so you look at this sentence to, which it says here, put a tick in one box that contains correct information about one of these fatty acids. So just one of this that contains the right fatty acid. Now that the correct statement corresponds, however, it also has to agree with this table. Okay, so it says caprylic acid is unsaturated, which means, pause the video here and try to attempt the question like I would normally say. So if you've done that, this is the answer. Caprylic acid is unsaturated fatty acid represented by the diagram L. So it says unsaturated. That means it should have one double bond or two double carbon to carbon double bond. We can't see any of that, and it says here too that it doesn't have double bond. Okay, so that is wrong because it's unsaturated means it should have a double bond. Linoleic acid is saturated fatty acid, which means that it should not have any bond. It should not have any double carbon to carbon double bond. So here, from this is N, this is E here. Now it says from this diagram there is no double bond, but from what we have on the table, it says it has two double bonds because the number of double bonds in the R group. So this is telling us that it does really have double bond. Maybe there was a mistake here or something. So this is also wrong because of this table. From this diagram it seems correct, but from this table it's not correct. So that we can't take that as well. It says palmitoleic acid is unsaturated fatty acid, which means you have at least one double bond in K. So it has one double bond, and here on this table here it shows it has one double bond. So definitely this will be your answer here. Excuse me, that's the answer there. That's why I choose here. Okay, stearic acid is saturated fatty acid represented in M. Well, this is wrong because you can see the two double bonds. I hope that made sense. Okay, now the next question says describe how ester bonds are formed in the phospholipids. Pay attention, the two kinds of phospholipids now, not triglycerides. Okay, so try to think about what the answer is and do let me know. And from what we just said, I think I want this to float again. Okay, one sec. Sorry, I want to just get that right. Okay, so sorry I had to do it that way. I literally splitted the screen because of the initial question. So this is a condensation reaction between glycerol and 2-fatty acid. Pay attention to that word, 2-fatty acid. Water molecule is produced in the process. You can also add that phosphate group is also added. That's fine, but this is enough for you to get your two answers, to your 2 marks. The diagram represents a triglyceride, so name the group represented in the diagram by the box P and Q. You can see that the Q is the same, so that should give you a hint into something. So let's see the answer to that. So your P is the glycerol and your Q is the fatty acid. Okay, I hope you didn't get to the next answer already. Name the type of bond between P and Q. This bond, what's the name? I guess you saw it already. Ester bonds. Okay, the next question here says describe how you would test a lipid sample in the presence of lipid and a liquid sample, yes, and how you would recognize a positive result. 2 marks. So this, when you say 2 mark, I miss no stories. So here you go. So the sample is dissolved in ethanol, then distilled water is added and shaken. If white emulsion, please not precipitate, emulsion appears, then lipid is present in the sample. Just straight to the answer. Okay, this is more like a 3-marker question anyways, but it's okay for you to write this. Like I said, there's nothing wrong writing more than required so that you have an insurance question or an insurance answer. So this question 5 is describe how you would test a piece of food for lipid. Basically just what we just did recently, literally. So it's the same thing. I literally just copied that and pasted that here. the sample is, because this is a past question from different past paper, so that's why it's repeated. So the sample is dissolved in ethanol, distilled water is added, shaked. If a white emulsion appears, lipid is present. Okay. Then this figure shows the phospholipids, then X and Y. So the question is saying here, the part phospholipid A. So please pause the video here to study this a little bit, please. So the part of lipid A is formed from a particular molecule. Name the molecule from lipid A, this part. So what molecule do you think this is? And that would be your what? Glycerol. Okay, then it says name the type of bond between A and X. Where's A and X? Between A and X. Same that between this A and X, this one. Well, you can say between A and Y as well, because the same bond holds them together anyways. So that is going to be what? Your ester bond. Then what? Let me stick to the question there. It says, which of the fatty acid X or Y is in the figure above unsaturated? So look at the diagram again. Which of them do you think is unsaturated? How did you see the whole length? And the answer should be what? It should be Y, because why will it be Y? Because Y has one carbon to carbon double bond. That's very important for you to remember. It has one carbon to carbon double bond. This is very important. Okay. Now, the last question here, I believe, says, scientists investigated the percentage of different types of lipid in plasma membrane from different types of cell. The table below shows there is also cholesterol in the lining of the lumen of mammals, in red blood cell in Escherichia coli. So you're meant to look at the number of each of them, so to say. So the scientists expressed this result as percentage. Explain how they will find this value. So how will you find for each, for the percentage of lipid plasma membrane by mass? So in who now? Plasma membrane. Okay. Like all of this. So how will they express it for each of the organisms? Like how would it be done? 2 markers. Read the question again to further understand it. So this is going to be done this way. So this can be done by dividing each mass by the total of all of the lipids. What does that mean? For example, you take, 17 over, so if I want to get one of cholesterol now, so just 17 over addition of all of them times 100. That's what that means. You can say that. So like this. So if it's for lipids, it's going to be 54 over all of these times 100, just like that. Okay, and that concludes that on lipids.